Now, this video, we're going to talk about our Form 4 Chapter 9.1 Bio AMALI, which is presence of amylase on the digestion of starch. First, of course, you need to know amylase is the enzyme that digests starch into maltose. Now, by the end of the experiment, right, how am I going to know whether starch is being digested? So there are actually two tests that you can carry out: Iodine's test and Benedict's test. Now, in this PDF, I got prepared notes for both tests, which you can download with the link and the description below. Now, sometimes they will ask together, sometimes they will separate us. So it's either iodine or Benedict, which I think they will separate because it's quite hard, quite time consuming to do both together. So first, you know, iodine and Benedict test what? So iodine is to test the presence of starch. So when there is starch, your iodine will turn from yellow to dark blue. So let's say if your starch is being digested into maltose already, your iodine will not turn dark blue because there's no more starch already. Now, Benedict's test is to test the presence of reducing sugar, like we learned in your Form 4 Chapter 4. So maltose is an example of reducing sugar. So other example of reducing sugar, there are 5 in total. 3 of your monosaccharides, the girlfriend gay one, glucose, fructose, galactose, and 2 of your disaccharides, maltose and also lactose. So when maltose or reduced sugar is present, your blue Benedict solution will form a brick red precipitate. So when your starch digests into maltose, you will get this. So if starch is not digested, then there's no brick red precipitate form, you will remain blue. So first, let's briefly go through this one first. When you get your paper, right, if let's say if it's an unguided experiment, there's no procedure, right? Okay, you have to look through the apparatus and materials and then look out for iodine or Benedict. If it's iodine, means it's iodine test law. If it's Benedict, then it's Benedict test law. If it's both, then you probably need to do both, which I don't think they will ask both lah. Now, after the list of apparatus and materials you write, the next thing I want to do is read the question. Must read the question. For especially this sentence, they will always write this one: to study, to identify, to investigate the effect of blah blah blah. So this is the aim of your experiment, because if you don't know the aim of the experiment, you don't know what you're doing, and most importantly, you don't know the variables. So they say here, to study the effect of presence of amylase on starch digestion. So presence of amylase is basically your MV. And then got amylase means got digestion. No amylase means no starch digestion. So starch digestion is your responding. However, later in your question, responding, you cannot write this, because you must write experiment when you're responding. Means how do you know starch is being digested? We get to that in a moment. Now, procedure I will go through later. I'll teach you how to write if it's unguided. So briefly, what you need to do here is there are 2 test tubes here, X and Y. One is starch and amylase. One is starch and distilled water. So the one with amylase will of course digest the starch into maltose. The one with distilled water will not digest the starch into maltose. So at the end of the experiment, you need to drop iodine to test the result. If it's Benedict, then of course we put Benedict, which is a bit different. I'll explain later. Okay, let's go through the variables first. So question usually variables, they'll ask like that one: state the variables and the method to handle variables. Now, method to handle, you must know if they ask you procedure, because for procedure, you must write this. But sometimes you will ask the question, like state the responding and then method to handle. State the constant, method to handle. Okay, first, MV, we already know, presence of amylase. So, whereby the 2 test tubes, right, one got amylase, one don't have, which is distilled water. So later when you write your procedure, right, so long you mention amylase and distilled water, you get marks for this already, for the manipulated variable. Responding, just now we mentioned it's the starch digestion. But in the experiment, how am I going to know starch is being digested? Well, I need to observe the color of the iodine or Benedict solution. So therefore, the color change will tell me whether there's digestion. So color change of the mixture in both of my test tube. So when you write your procedure that time, you must mention this: observe and record the color change. Okay? Constant variable is your temperature. So you need optimum temperature for enzyme to function, for amylase to function. So both test tubes must be immersed in 37 degrees water. So temperature of water bath, fix the temperature at 37. So when you write 37 degrees, right, the value of the constant variable, you get marks for this. Okay, so now, how do I write procedure if it's unguided? So procedure, there are 4 things you need to write. 3 methods to handle the variables that you just mentioned just now, and 1 precautionary step. Precautionary step means how do you improve the accuracy of your experiment, right? These 4 only will give you marks, the rest no marks one. But you still need to write the rest to complete the procedure lah. So first, both test tube X and Y, I add 1 ml of 1% starch suspension. Okay, I put starch first. Then, the next thing is different already. Test tube X, I put 2 ml of 1% amylase. And the other one, I filled with distilled water, 2 ml also lah. So long you mention amylase and distilled water, you get marks for the manipulated variable. So this one I get 1 mark already. This one got marks one. Then next is the precautionary step. For this experiment, you need to use glass rod to stir the mixture, because if you don't stir it properly, the amylase, might not react with the starch properly. So stir the mixture well using a glass rod. This is your precautionary. So you make your experiment more accurate. Then I put both test tube in the 37 degrees water bath. So long I mention 37 degrees punya water bath, that is your constant. Then I start stopwatch. So probably you need to soak around 10 minutes lah if it's unguided. If it's guided, just follow whatever time they give you. After 10 minutes, then I will do the iodine test. So I'll put 3 drops of iodine solution in each test tube, and then you'll get your results. So you observe and record the color change of the mixture. So that is your responding. Responding is always at your last point one, because your observation is your responding, which is always at the end of the experiment when you get your results. Okay, that. But if it's Benedict solution, okay, a bit mafan, a bit more steps. Let me show you here. So there's more things you need to do. Okay, so after 10 minutes, right, we don't still add the Benedict like we add iodine. We need to remove 2 ml of mixture from each test tube, put it into another test tube, and then you need to heat it in a, you add the Benedict, then only you add the Benedict solution into this different test tube, and then heat the water bath. Because Benedict solution need heat to react around 100 degrees Celsius. So after 10 minutes, I remove 2 ml of the mixture from both boiling tube and put it in 2 different test tubes, and then you add 3 drops of Benedict solution into here, and then in a boiling water bath for 1 minute. That's why you need Bunsen burner to heat this Benedict one. Then you get result. Okay, then same thing, you observe and record the color change. So for Benedict's test, right, the variables are the same. You just use different tests on it. Because for Benedict, you also need to observe and record the color change. The color change is different. Once you've done that already, the next question, obviously they will definitely ask this one, observation and inference. What do you see in the end? So for iodine and Benedict's test, right, your observation obviously is different because the color change is not the same. So for iodine test, right, now test tube X and Y, we have different contents. So test tube X is starch and amylase. The other one is starch and distilled water. So the one with amylase, right, will of course digest the starch into maltose. The one without the amylase will not digest the starch. So when I put iodine into test tube X that time, iodine to test presence of starch. Now no more starch already. So your iodine initially yellow, at the end still remain yellow. For the other one, starch is not digested. So in the beginning of experiment got starch, in the end still got starch. So your iodine will change from yellow to dark blue, because when starch is present, iodine will change from yellow to dark blue. Then inference means explain your observation. So why you will get this result? So why when I put iodine here, I remain yellow? Because no more starch. Starch is digested. You write this actually enough already, but here I give you one more mark point explanation, because now starch digested, there's no more starch, so iodine doesn't change. So the other one is starch is not digested, so you still contain starch. That's why your iodine will change to dark blue. For Benedict's test, of course the color change different lah. So for Benedict, test tube X got digestion, means your starch become maltose already. So your Benedict will change from blue to brick red precipitate. But for the other one, your starch is not digested. So if there's no starch digestion, there's no maltose, no reducing sugar form, Benedict solution will remain blue color. And for the inference, actually it's more or less the same. Like for here, I just say, why here the Benedict will change color? Because starch become maltose already, no? And here I give you one more point also, better to write this also, because maltose is a reducing sugar. Benedict, when got reducing sugar, will change color. change from blue to brick red. For the other one, starch not digested. So starch doesn't form the maltose, the reducing sugar. So your Benedict will not change from blue to brick red. There's no reducing sugar present. So for Benedict and iodine test, right, only these two questions is slightly different. The rest, more or less the same, right? Even for the variables that we just mentioned earlier. Right, moving on, we do hypothesis. Now, in most experiments, right, hypothesis, we always can say 什么跟什么跟越越, the higher the MV, the higher the RV. But for this experiment, you can't do that. It's because this MV, right, is not something you can measure. It's not a value. It's not like there's cm, percentage, mole, concentration like that. You can use number, 1.1, 1.2. No, it's whether got amylase or not. So for this kind of experiment, you cannot say higher the amylase, higher the color change. For this kind of experiment, hypothesis will go back to your aim. So you need to answer the aim of the experiment. Just now the aim say presence of amylase affects starch. So you just have to say, look, when got amylase present, starch is digested to maltose. That's it. Or you want to say, when there's no amylase, starch is not digested into maltose. So answer the aim for this kind of question. So when amylase present, starch become maltose. Okay? And the next question is more or less the same like inference hypothesis like that. How does the result of the experiment show the starch digestion has occurred in the test tube? X. So the result here, how do you know this one will show digestion? Because now you just mentioned, the iodine remain yellow. Iodine remain yellow means there is no more starch. Starch already digested by amylase into maltose. If it's Benedict's test, you just say the Benedict's form brick red precipitate. So this Benedict solution test form brick red precipitate. So you just mention the result. Because if there's digestion, you get maltose. Maltose is reducing sugar, then you get brick red precipitate. Understand? And finally, a few KBAT questions here, which quite important. You might want to listen. Now, a student carried the same experiment by using Boyd amylase solution. Predict the outcome of this experiment. Now, this could be a possible experiment they could ask also, which instead of using presence of amylase as MV, right, they could use temperature of water bath as your MV. Okay, so they might change a bit. So I give you another possibility here. So if let's say they ask you to use 100 degrees 100 degrees Celsius punya amylase. So let's say now your MV, right, is temperature. One is 37. One I soak it in another temperature, 100 degrees Celsius. So 100 degrees Celsius will cause, okay, and then this one is amylase also, of course. Now 100 degrees Celsius will cause the amylase to denature. Means this one will also won't have reaction. This one got. So which means iodine, if you put here, this one remain yellow, this one dark blue. For Benedict, same also. This one brick red precipitate, this one remain blue. Basically, you put hot water, there's no reaction, just like the distilled water, same one. So this is a possible question they can ask. Now, if they ask like this, right, MV then will be temperature already. Temperature of water bath. Responding will still be the same, but constant takkan you write temperature again. So constant will be your enzyme and your substrate. So you can say concentration of starch, concentration of amylase, which 1% or whatever value they give you in your paper lah. Understand? Okay, so for this one, they just like ask you to predict, or if I use Boyd amylase. So if I use Boyd amylase, dark blue color solution will be produced because now your starch is not digested. Okay, so when starch is present in the end, iodine will test the presence of starch, so you form dark blue. So the reason I told you already, enzyme denatured, no starch can be digested into maltose. And then the last one here, KBAT question I took from reference book 1. Okay, it's about digestion, like your digestion knowledge they test you. A woman took a lot of unripe mangoes before eating rice. Predict the hydrolysis of starch in the duodenum. Now, in your duodenum, the pH is alkaline. So amylase enzyme in your duodenum requires alkali condition to function in your duodenum. But unripe mangoes is acid. So if you eat too much acidic food, the acid will affect the alkali condition in your duodenum. So therefore now the amylase cannot function very well. The digestion is not efficient. So that's why here I say already, less starch is hydrolyzed, is digested to maltose. The reason is because the pH not ngam already. So at the duodenum, I mentioned here, amylase reacts optimally at alkaline condition, which is 8.2. But you can just write alkaline good enough already. But here's the main thing. Just mentioned, mango is because they are acidic. It will change the alkali condition in your duodenum. So the alkalinity in duodenum reduce. So rate of amylase reaction also reduce. So the enzyme doesn't function very well. Alright, so I hope that this video helped you guys a lot on your upcoming AMALI. Hopefully it's the same. And if you got any questions, please ask in the comment. And I'll see you guys real soon. Bye-bye.